Well, I'm part of the directorate, so I've been here from the beginning of WIN, and I'm constantly working on trying to help it succeed. My particular project is trying to solve a very old problem, a key problem in cancer therapeutics, using some new precision medicine orientation. What happens inevitably to the majority of cancer patients is that they relapse, they have metastatic disease, and then they start treatment. For some diseases, the treatment is very, very effective, but always just for a limited period of time. Inevitably, the patients relapse. And then they ultimately, regardless of the second and third lines of therapy, they ultimately die of resistant metastatic disease. So the aim is to overcome resistance to therapy. And over the years, we've identified specific mechanisms that result in resistance, but there are multiple mechanisms. And there's been no way of targeting all of them, and also no way of targeting all of them and not in normal tissue. In other words, not sensitizing the normal tissue and enhancing the toxicity to the patients to the point where nothing's accomplished. It turns out that we've identified a transcription factor that regulates a whole host of factors that protect the tumor cells, cytoprotective gene battery. It flushes the drugs out, it metabolizes them, it turns on other pathways that favor the tumor cell. And this is called NRF2. NRF2 is regulated by another protein called KEAP1. And as it happens, KEAP1 is mutated in a variety of human cancers, up to 30% of lung cancer patients, but in a variety of other cancers. When the KEAP1 is mutated, it can no longer bind NRF2 and take it to its destruction to maintain it at a low level. Consequently, you have a constitutively elevated NRF2 resistant tumor cells. And this is somatic mutations. This is only in the tumor. So using molecular modeling and drug design, we have found a drug that corrects the mutated KEAP and reestablishes its binding with NRF2. And so it lowers the NRF2, sensitizes the tumor cells and the tumor cells selectively, because it's only the tumor cells that have this KEAP mutation. So now we have a selective way of lowering resistance, sensitizing tumor cells to not only cytotoxic chemotherapies, but potentially also immunotherapies. Resistance to immunotherapy is a real big problem, and targeted therapies of various types. More than that, it's only in patients, obviously, that carry this mutation. So we can now use the same paradigm we've been using, take a tumor from the patient, look at KEAP. If it's mutated, we can choose this drug, which will sensitize the tumor and only the tumor to whatever the other treatment is. It turns out that no one drug, there are many, many mutations in KEAP, no one drug covers them all. We found a second one that covers not all that the first drug does, but a different one. So actually we're going to have a customized, depending on where the mutation is, which is easily done in a molecular test, NGS screening, and we'll choose the right drug. So we're just optimizing the drugs. We've done all the preclinical animal models. So now we've just gotten some funding in Canada to take it to preclinical talks, and then we'll be looking for a partner to develop this clinically.